Bonjour, year 9. Welcome to week 5, lesson 21. This is about digestion and making digestion more efficient. So before we begin, make sure you understand the word digestion. So that means making large insoluble molecules into smaller soluble ones. So how we get large molecules such as carbohydrate and we break them down into smaller soluble substances. So large carbohydrates can't dissolve into your blood. Smaller soluble molecules such as glucose can. So it's about taking those large insoluble molecules and making small soluble ones that we find useful. So for example, that glucose is used in respiration. Let's look at this diagram. It makes me feel sick a little bit. But this is a beautiful image of the inside of the small intestine. All those little finger-like projections, they're called villi, and just sort of imagining them waving along as the gut moves. I find this a very satisfying image, whilst very disgusting. So let's talk about today making digestion efficient. So let's have a little think about this. So you have more bacteria cells in your body than you do body cells. You are more bacteria than you are anything else. When you poop, that's more bacteria than anything else. So let's think about how useful bacteria is in your body. Now, we tend to think of bacteria as being a bad thing. When they are a pathogen, so when they make you poorly, they are a bad thing. However, you are majority bacteria. So gross, but we need to think about how is bacteria useful. So think about these 3 questions. How do gut bacteria aid digestion? So what do they do? How do gut bacteria help the immune system? And what are probiotic supplements? And do we need them? So there's a picture of Yakult there and what's the other one called? Actimel. What are they and do we need them? Give it a little pause while you think. Okay, hopefully you've had a little bit of a question there. So gut bacteria does a lot of things. So it communicates with your immune system. It also helps you to produce certain nutrients and vitamins that you require. They also help to digest down the food. They help promote sort of the environment for good digestion. Probiotics also provide either new cultures or they provide good conditions for cultures to flourish within the gut. So communicating with your immune system and helping break down matter and making new matter, which is really, really impressive. So we need to understand what makes digestion more efficient. We're going to look at bile, we're going to look at hydrochloric acid, and we're going to go for those 7 and 8 grades. So we're going to be able to very easily explain how acid in the stomach increases the efficiency of pepsin, which is an enzyme, and we're going to talk about how bile increases the efficiency of fat digestion and how we can see that to be true. Okay, so this is a 3-minute challenge. For each part of the sandwich, you've got to name the enzyme type and the type of enzyme that helps to digest it and what it's broken down into. For extra challenge, can you name the active site and the substrate? I hope you've had a really good pause there. I can't remember what's on the next slide, so it might be that I'm going to go through all of it. Okay, so ham sandwich. I'm just going to focus on the bread initially. So bread is made of carbohydrate, starch, and carbohydrates such as amylase will break it down into glucose. The cheese has got a lot of fat in it, and that fat will be digested by lipases and with the help of bile into fatty acids. Soluble, we can use fatty acids. The ham is going to contain a lot of protein. Protein is broken down by the enzymes proteases, and then that is going to be breaking it down into amino acids. Quite a lot of acids. Obviously, within the rest of the sandwich, you've got a lot of water, and you've also got some other vitamins and minerals, but they're the 3 main constituents. What is an active site? The active site is the place where a substrate will attach to an enzyme. That is where the magic happens, so to speak, in the enzyme. And the substrate is the name for anything that is being broken down by an enzyme. So if we remember our lock and key hypothesis, if you haven't done that yet, you might want to go back and have a look at the way enzymes work. Okay, so enzymes are biological catalysts. If you didn't know that already, write that down. They speed up chemical reactions in living organisms. They do this by lowering the activation energy for the reaction, minimum amount of energy required for a reaction to occur. Massive link here to C2? I think so. So without enzymes, we couldn't digest our food. We couldn't break down those large insoluble substances into soluble ones, and so we wouldn't survive. It would take years to break down complex foods like a ham sandwich. Every enzyme is very specific, so each enzyme will only break down or will only catalyze a reaction where we have a specific shape substrate. So enzymes are made of proteins that are sort of coiled up into specific shapes, and they match a certain shape. So for example, glucose and carbohydrates are made of chains of like hexagon shapes, and so the enzyme active site would be a hexagon shape, and that would fit in perfectly like a lock and key. Optimum temperature for a human enzyme, have a think. Shouldn't be too much thinking time because it's body temperature, about 37 degrees. Boom. We did an experiment on optimum pH as well. So we have to keep the pH of our digestive system. It's complicated because we have different parts of our body that have different pHs. We know that in our stomach we have acids, so that's going to have a low pH. In our mouth, slightly alkaline, so it's going to have a higher pH. So we have lots of different chemicals providing lots of different conditions for the lots of different enzymes that work at different pHs. Very complicated throughout the digestive system because we have lots of different conditions that are suitable for certain enzymes. Can you think of any other substances involved in digestion? Well, I've kind of said about the stomach acid, but in your mouth, what have you got in your mouth? You've got saliva that lubricates your food and enables the enzymes to be mixed in thoroughly. We mentioned acid. We've also got this beautiful green stuff. This is called bile. People kind of think of bile when they think of being sick, but bile's a really, really useful part of digestion. So the stomach, where we have mostly the digestion of proteins, we have a specific enzyme called pepsin, which is a protease. So a protease is an enzyme that breaks down proteins, and it's secreted by glands in your stomach. So pepsin breaks down lipids into fatty acids and glycerol. It works best in acidic conditions, which is why we have hydrochloric acid concentrated in our stomach. It kills any bad microbes in our food. You produce 3 litres of hydrochloric acid a day. Wowzers. So here's a picture of the lining of the stomach and the stomach acid there. So just to show you here, we have got a piece of food in hydrochloric acid, a piece of food in pepsin, and you can see when we've got pepsin and hydrochloric acid together, that piece of food has been dissolved or digested because the two complement one another. They work best within, so pepsin works best within acidic conditions. Your stomach produces a thick mucus which protects them from being digested from your own acid and enzymes. So if you perforate the stomach lining, for example, you're going to be in real trouble. So that mucus protects you. And if you have an ulcer, the mucus is lost, and so the acid, the lining of the stomach becomes attached, and it's very, very painful. It's a chemical burn from the inside. Yummy. So for your notes, this is the key stuff here. We need stomach acid because it kills bacteria in your food. So it protects us from things like E. coli, salmonella, obviously if you take in board too much of that. So this is one way our stomach acid helps our immune system. It denatures proteins, so it makes the digestion easier. It allows us to break down proteins better. It activates your stomach enzymes that only work in specific conditions, and pepsin is inactive when it's first made. It's only activated when acid is added to it. We're not going to do this because we did this last week about investigating the effect of pH on enzymes, but you can have a look at this on Kaboodle if you wish. So this is the student activity of investigating the effect of pH on enzyme activities. If you go back to the last one of last week, you'll be able to see the investigation with the effect of pH on enzymes. So we've eaten our food, nom, 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 nom. Carbohydrates have been broken down into maltose. Carbohydrates have been broken down by carbohydrates into maltose. We've swallowed them down our esophagus. They've gone into our stomach, where pepsin has broken down the proteins into amino acids. And then when we go between the stomach and the small intestine, we have more enzymes added to the food made in the pancreas and the small intestine, producing alkaline conditions. So we're now neutralizing that stomach acid because, again, we don't want it coming into contact with our tissues. We also produce bile. Now, bile is made in the liver, stored in the gallbladder, and is injected into our food. It neutralizes the acid of the stomach. So you need to know the two things that bile does. So the first thing is it neutralizes stomach acid. And the other thing is it emulsifies fat. If you increase the surface area of fat, it means enzymes can digest them much easier. So emulsify means spreads out into smaller droplets. Bile is really, really useful. Everyone produces it. If people have gallstones, it means that those stones are getting in the way of the bile being injected into your small intestine. So that's where pain can be caused. Lesser known fact about Mrs. Corbett, she does not have a gallbladder. Like a horse. She eats like a horse too, so bad times. Okay, so here's a picture of the liver, and then you can see that it's produced in the liver where everything's green, goes through the hepatic duct into the gallbladder. All you need to know is it's made in the liver, stored in the gallbladder, and then it goes into the duodenum, which is the very first part of the small intestine. So it's kind of injected in to neutralize that acid, but also emulsify fat. So the enzymes work better if it's a rate of reaction. We know that if we have got a larger surface area, so smaller droplets, we can more efficiently increase the rate of reaction. So fats are soluble. Fats are insoluble in liquid, so they kind of form big globules, whereas bile spreads out the fat globules, and then the enzymes can go in and do their thing much more efficiently. So they can break them down into fatty acids and glycerol much easier when they're in much smaller droplets. It's kind of like, I always think, if I had to eat a giant Malteser that's the size of 100 Maltesers, or I had to eat 100 individual Maltesers, I could probably do that much, much quicker because they have a larger surface area, because I can pop them in one by one, instead of having to bite into this massive sphere of Malteser. Really want some Maltesers. Okay, here's an activity. You will need to pause for this one. Read through both of these methods. And both of these methods are showing how bile works. So interpret the observation for each one. What does it show about the function of bile? And B, explain how each function makes digestion more efficient. Have a read. Oh, I hope you had a good think about those. So interpreting the function of data of each one and explaining how it makes it more efficient. So make sure you have a purple or green pen to mark your work. You don't obviously have to. No one is going to check right now, but tick your work. Makes you feel good to get things right, and correct it if you get it wrong. So experiment 1 showed bile breaking down fat droplets from large droplets into smaller droplets, showing that it emulsifies fat. It increases the surface area, so it means that lipase can work more efficiently to catalyze the breakdown of the lipids. Experiment 1, is more qualitative because there's no data. Experiment 2 is quantitative because we can see a change in numbers. So bile is more alkaline than water because the pH is higher. Bile neutralizes the stomach acid juices so that enzymes in the small intestine can work at their optimum pH, which is slightly alkaline, not acidic, as it's just come from the stomach. Like 4 points. Give yourself some ticks and some smiley faces. Here is a 6-mark question, slightly different. Describe how different parts of the human digestive system help to break down molecules of fat so that they can be absorbed into the body. Make references to any enzymes involved, the products of digestion, and any other chemicals involved in the processes. Now, this is a 6-mark question, so you're going to need to take a minimum 6 minutes to do it, really. I always say a point per mark. It says 15 minutes in the corner of the screen. You might need all of that initially, but probably not to the end. Now, this is a really complicated question because it has 2 large parts. So it's got 2 sentences. Describe how different parts of the digestive system help to break down molecules of fat so they can be absorbed into the body. So we're only interested in fat here. So I would circle that. Make references to enzymes involved. Circle that. Products. Circle that. Other chemicals. Circle that. So there's 4 things really we need to look at. So pause it, think and see if you can answer that. So I would start with, what part of the digestive system is fat broken down in? What are the enzymes that do it? What do they make after they are fat? What are they broken down into? Is there anything else that helps the enzymes? Okay, so I'm not going to bother. You can mark your own. I'm going to start with the 5 to 6 marks. So if you have written, well, this one starts with the mouth, obviously. So the mouth and the stomach increases the surface area. What I'm mostly interested in is that fat is predominantly broken down in the small intestine. It's predominantly digested in the small intestine. Lipase is the enzyme that does it. And it breaks it down into fatty acids and glycerol. So I would say tick small intestine, tick fatty acids and glycerol, tick lipase. The other chemical, bile, is produced in the liver, tick, comes from the gallbladder, tick. And the 2 things it does, it emulsifies fat and it neutralizes stomach acid, tick, tick. That's your 6 marks. You would then say that the fatty acids and glycerol are small and soluble, and so they can go into the bloodstream. Let's talk about pepsin. So that we've already sort of spoken about pepsin. It's found in the stomach and needs a low pH, so it doesn't denature. Trypsin is found in the small intestine, which has alkaline conditions, and therefore the optimum pH needs to be higher. So pepsin has a optimum pH of 3 and trypsin has an optimum pH of 8. Describe the different pH conditions required by pepsin and trypsin and how these are achieved. Well, we've already spoken about pepsin. The stomach produces and secretes pepsin and stomach acid to allow it to have those lower pHs. The trypsin works with a pH of 8, and so we know that bile increases the pH of the small intestine, doesn't it? It neutralizes that acid. This is something that you can choose to do at home. Explain the journey of a burger through the digestive system using what you've learned from last week as well. Explain your ideas. Could be a cartoon or a story or a picture. If you've got some artwork you can send me, amazing, please do. My wall's looking bare. I am printing a lot of the stuff that people have sent me just to kind of keep me motivated. Like, these are the things that people are making at home. Any kind of art, really. I enjoy art. Oh, there's a burger. There's another burger. Double burgers. Here are the grade boundaries, so you'll know if you are dealing with our grade 6s and 7s, so I'm going to focus on that. All of the organs in the digestive system, structure, shape, function of the organs, what happens to the food in each organ, what we use all of the nutrients for, so fat, proteins, sugars, all that kind of thing, how enzymes work, how they're involved, how the food is digested, absorbed and transported to the cells. Don't bother with the 4 and 5, go for the 6 and 7. Because you're brilliant. And we're done. Have a look at that, 7 to 8. Can you do all of those things? Can you explain how stomach acid increases efficiency of pepsin? Can you explain how bile increases the efficiency of fat digestion? Well, you should be able to. Worth noting, there is also another video that goes with this, and this is looking at the model gut, what it is, how it works, because quite often they put these things in and students get confused. It also really nicely links osmosis, diffusion, and the food tests for iodine and not for iodine, that's stupid, for carbohydrates such as starch and for sugars such as glucose. Go check it out. Bye.